Hi everyone, we're back talking about arthropods. So welcome to the arthropod YouTube video. So we recently discussed the phylum Annelida, and now we're talking about phylum Arthropoda, which you've all been working on as you collect your insects for your insect collection. So there's a bunch of animals that you probably recognize on the screen in front of you because arthropods includes lots of different organisms that are well known. So insects, crabs, shrimp, spiders, scorpions, copepods, which are these fun little guys down here, all kinds of crustaceans and insects, millipedes, all kinds of fun things. So lots of things in this phylum that you've heard about before. Okay, so this is organized into 5 subphyla, and arthropods, this is where it gets a little tricky, right? Because as things get learned more and more about, we learn more about these subphyla. So in addition to having a phylum and the classes, we're also going to have subphyla in here. So you're responsible for knowing these 5 subphyla. And these are Chelicerata, which includes spiders, scorpions, and horseshoe crabs. Crustacea, which is shrimps, real crabs, lobsters, and copepods. And note that horseshoe crabs are in Chelicerata, not in Crustacea. And so we'll talk about that a little later on. But essentially, all the things that you eat, most of the things that you eat, are going to be crustaceans. Hexapoda is another one. Myriapoda, which is going to be your centipedes and millipedes. And Trilobitomorpha. And this is an entirely extinct subphylum, but again, very well known because trilobites were part of a lot of fossil records. So we'll know about them through that. Okay, so here is a showing you Arthropoda and showing you the 5 subphyla nested within this phylogeny. And also note out here, right? So tardigrada is now our sister taxa to all the arthropods. And then Oncophora, which is sometimes a group that's closely related to these arthropods, is here too. And so we haven't really talked about them much, but they're also out here as well, a different phylum. Okay, so these 5 subphyla used to be their own phyla, but now are lumped under Arthropoda. And the sister taxa to Arthropoda is Tardigrada, which we already talked about. Okay, and so you'll see mapped onto this phylogeny all the things we're going to talk about over the next couple slides. And included things like jointed legs. So our outgroup for arthropods is going to have unjointed appendages. Tardigrades, including in this group, are going to have jointed legs. And then you start getting into segmentation more when you get into all the arthropods. So we'll talk about that coming up. But what you'll note about all these groups is that one of the traits here at the bottom of this tree is these ecdysial glands, which are for ecdysis, which again is going to be that molting. So we saw them molting in tardigrades in lab. We saw molting in arthropods as well. So here comes. Okay, so here we are, ecdysozoa, things that molt their exoskeleton. Those includes arthropods, nematodes, tardigrades, right? And this is compared to this other invertebrate supergroup, Lophotrochozoa, which includes annelids, mollusks, platyhelminthes, rotiferans, all these things that are grouped together based on their RNA profile. So based on genetic information, not on physical information. Okay. All right. So arthropods are a very successful group, 30 to 50 million species. And just like we've seen so far in our recent animals, right? Triploblastic organs, bilateral symmetry, open circulatory system. So their blood is, like in most mollusks, is going to be in open vessels, not in closed vessels. So not in blood vessels, but in open sinuses. Okay. The coelom is there, but it's pretty reduced. And they live all over the place. And you've noticed this when you're catching your insects, right? You've got marine, freshwater, and terrestrial insects. Okay. So if we were measuring success by number of species, arthropods would be the most successful. So it's the biggest phylum, the most diverse phylum of all the phyla we'll talk about, all the phyla of mammals. And there's been 4 major things that we think have helped with arthropods be successful. So this is going to be metamerism and tagmatization. We'll talk about that. Exoskeleton, the hemocoel, and metamorphosis. Okay. So in metamerism, we saw this in the annelids, right? We saw that there's externally evident repeated segments, including appendages. In arthropods, actually, a lot of the internal segmentation has been lost, so the internal metamerism. So there's actually not quite as much of the repeating organs as there was in annelids, okay? They still have it. And this could be the result of having exoskeleton. So in annelids and other metameric phyla, those animals are going to have a hydrostatic skeleton, whereas these ones are going to have an exoskeleton, so different kind of support system here. Okay. And tegmatization is going to be regional specialization and fusion of those segments. Okay, so they have segments and then they have fusion of segments. And we see this really nicely in the arthropods because if you look at something like a lobster, you see that they have all these different segments with multiple legs on them. And then once you have all these legs, now you can specialize. You can have legs that swim, legs that walk, legs that grab, right? So it gives a lot of options then for these animals to move and to feed and to reproduce. So highly specialized body regions we see in arthropods. And we've got tagmata, so regions that are for feeding, sensory perception, locomotion, all different kinds of things like that. Okay. So the exoskeleton is something else that we think really has helped arthropods be successful. And their exoskeleton is strong and light and also has external joints. So it's really successful and works really well for them. And this is cool because the exoskeleton covers all the body surfaces and the invagination. So this beginning part of the mouth and the pharynx and the end of the hindgut is still all covered in exoskeleton. So that part's crunchy too. Lots of protection then, really good evolutionary strategy. And the exoskeleton is non-living. So the hypodermis, a portion of the skin, is secreting this non-living exterior, which means that you're not going to be having pain receptors out there. You're not going to be having damage, right? If you're damaging out the exoskeleton, you're not damaging the living tissue. So it's a really good strategy for that as well. Okay, so this is sort of interesting because now we have external joints. So we have internal joints, right? When our bones are joined together, we have a joint here, say in our elbow, and that is going to be an internal joint. But with the external joints now, the muscles are actually inside of that, not outside like in ours. So the muscles are inside the joint and then pull internally to move the jointed exoskeleton. So it's a really, really cool system. And this provides structural support, protection. Exoskeleton is impermeable to water. So this is a really great strategy for reducing water loss. And in animals that are living on land, you need to worry about that, right? We've got really great muscle attachment points. So like our bones, but again, from the inside to the outside, we see attachment points for muscles on those exoskeleton pieces. Okay, so there's two layers of an exoskeleton. First is this epicuticle, which is a waxy lipoprotein. So a waxy, fatty protein substance on the top that essentially waterproofs them. Okay, so it's impermeable to water and also to pathogens. So it keeps them from getting sick. A good layer of immune protection there. And this underneath procuticle, and the procuticle is most of the exoskeleton. This includes that chitin that we've heard about some, which is tough and leathery. And it's hardened by a process called sclerotization, where when you tan the phenols in there, it creates these cross-links that make it a lot tougher. So sclerotization is a tanning process that chemically alters the structure of this procuticle, making it tougher. And then there's also calcium carbonate embedded in there. So they're crunchy. If you've ever crunched into a crab claw or a crustacean, you'll see exactly how crunchy they are, right? Step on a cockroach, you know this, they're really crunchy. Okay, so what this means though is that though it's really great strategy, they still have to grow somehow, and they can't grow if they are pushing against this crunchy exterior. So they have to go through ecdysis, or shedding that exoskeleton. And then for a little while after they shed it, they're going to be soft in order to grow, and then they can reharden the exoskeleton. So in order to grow, in order to do this, they go through something called pre-ecdysis, where a bunch of enzymes are secreted that digest and separate the hypodermis, so that underneath skin layer, from that non-living exoskeleton. Okay, so they're secreting this molting gel of enzymes here that separates the living from the non-living part, and then that can come off here. So then underneath that molting gel, we have a new epicuticle and a procuticle secreted, and eventually then that old exoskeleton is shed. So during this time here, once this is coming off, now your animal is soft for a while, okay? And your new exoskeleton is then inflated and it grows as the animal grows until it hardens. Okay. So after it hardens, after the animal has grown and now it rehardens, then you have further of the new exoskeleton sclerotization and this calcium carbonate secretion. Okay, so this allows for a lot of growth and metamorphosis too, right? So most organisms in arthropods go through metamorphosis, where we have two distinct forms of the same individual in life cycle. And so the most common example is going to be a butterfly, where you have the caterpillars forming a cocoon and then completely changing over body tissues into this adult form of the butterfly. And why this is a really great strategy, why we see it in so many organisms, is that the larva is going to live in a different place than the adult's going to live. So the caterpillar is going to live on our trees and branches and leaves and eat the leaves, and the butterfly can fly around and access nectar. So giving them more access to resources. It also reduces competition between the juvenile and adult. So this gives everybody more food available because the adults can feed on different things than the juvenile can. Okay. Another characteristic of arthropods that we'll see when we do our virtual labs is something called a biramus appendage. Okay, and this means just two branches. Biramus means two branches, and so you see that here. Here's this this thing called the protopod coming off the body wall, and then there's two bits to this. There is an exopod and endopod, so two branches. Okay, and the attachment point is called that protopodite. Okay, so you have an exopodite and an endopodite, meaning the inner and the outer branches of your appendage. Okay, so then our first subphylum of arthropods is our subphylum Chelicerata, and we'll watch this video together on Wednesday, but if you want to watch it beforehand, you can. It's pretty cool. There's 80,000 species of chelicerates, and this includes mostly spiders, but also includes horseshoe crabs, which is going to be class Merostomata, class Arachnida, spiders and scorpions, and then class Pycnogonida, which is the sea spider. So if you ever knew, there are sea spiders. Ooh. And chelicerates have two body tagmata, okay? So they have two major regions of specialization in their bodies, the cephalothorax and the abdomen, okay? And then they also have a pair of chelicerae, which Chelicerata is named for the chelicerae, which are these pinchers at the front, okay? And they have these pedipalps which help them get food in to their mouth as well. Okay. So it's thought that the very first land animals were your second subphylum, Myriapoda, and this lineages are thought to be around 428 million years old. So millipedes and centipedes are really old animals and very successful because they're still in the similar body shape we see in fossils. Okay. Aquatic scorpions are up to 425 million years old. So scorpions are going to be in that Chelicerata group, whereas terrestrial ones are going to be more along the lines of 400 million years old. Okay. And so this is a major strategy that we're seeing in arthropods, is that you see a lot of movement to land that requires water conservation, right? So if you're living in water, you're able to get water whenever you need it, even if it's salty. But if you're moving on land, now you really have to conserve water, okay? And so exoskeleton is an example of something called an exaptation, where that it was originally thought to have evolved for something else, okay? So it's evolved for protection, something like that. In this case, the exoskeleton then now is actually used for water conservation, which is a major thing on land. So we saw the exoskeleton evolving in the water as a protection mechanism, but now on land, it's a great strategy for reducing water loss. Okay, so I'm gonna take a break there, and we'll start with a new video, Arthropods 2, coming up.